In question set 18, we're going to talk about biotechnology and also finish our case on MSUD. Let's first define what biotechnology is. Whenever we look at any type of biotechnology, what we're talking about is some use of artificial methods, and the goal of biotechnology is to somehow modify genetic material. And this could be of a whole living organism, or it could be of just a cell. You know, we've been doing a number of things with biotechnology for many years. Biotechnology has been used to improve livestock. If we think about what we want from a cow in terms of milk production, or whether we want more muscle mass on a pig, we realize that biotechnology can be helpful with that. Additionally, when we talk about various types of crops, we know that some types of crops are what we term genetically modified. So genetically modified. There's been a lot of debate in the news of whether consumers should be made known to whether they're eating something that has been genetically modified. I suspect that you eat genetically modified foods every day, and you may not even be aware of that. We also can use biotechnology in other areas, especially in the area of medicine. When we've gone through the production of vaccines or antibiotics, biotechnology has been in use. We've also used biotechnology in cleaning up oil spills. And biotechnology has even helped us improve our enzymes for doing the laundry. More recently, there's a new gene editing type of technology called CRISPR. And I won't tell you what CRISPR stands for, it's a big long term, but essentially it allows us to do gene editing in humans. And this could potentially eliminate certain types of diseases, although it also has a potential for abuse when we think about perhaps designing babies the way we want them with certain quote-unquote desirable traits. So you may hear a lot more about CRISPR in the news. As we think about biotechnology, there's a couple of important things that we have to do in order to be able to modify genetic material. And one of the first things that we need to do is to be able to extract the DNA. So I wanted to go through a figure in your textbook that discusses the steps for how to actually extract DNA and how we can actually then manipulate that in the laboratory. So you have a place in your course manual where you can jot down these steps. The very first thing that has to happen if you want to modify genetic information is you have to get your cells and you have to be able to lyse them. Lysing them, I hope we remember, means to break them open. And the way that we do that is we use a type of detergent that's going to disrupt that plasma membrane. Remember, plasma membranes are made of phospholipids, and so if we have a type of detergent that actually breaks lipids apart, then this is the way that we can break that cell apart. That's the first step, because we remember that DNA is going to be located in the nucleus. The second step is that we have to be able to treat our tube, now with our cells that are broken open, with various enzymes. And one example of that would be something called protease. But if you just know that we have to treat this tube with enzymes, what we want to do is to prevent DNA from being degraded. So we want to prevent DNA from being broken down. And the way to do that is to add certain enzymes to the tube that actually is going to destroy some of the things that could break down the DNA. And so that's something that has to be done early in the process, immediately after the plasma membrane has been broken apart. The next thing that we'll do in step number 3 is to pellet our cell debris in a centrifuge. A centrifuge is a machine that is going to take this tube and it's going to spin it really, really fast. And what it does is it actually pulls anything that's heavy to the bottom of the tube. And in this case, that's where the cell debris is. And this cell debris then are bits and pieces of our plasma membrane and other types of organelles. And it's the part that we don't want. It's actually the trash that we want to get rid of. What that leaves for us on the surface, shown in this picture in this sort of yellow color here, is going to be the liquid that's left on the top that's not quite as heavy, and it's inside of this liquid where the DNA is going to be located. So what you'll need to do here is actually get a pipette. This particular picture used a glass rod, and they actually transferred that into a clean tube. Once that liquid is going to be in that clean tube, then the DNA needs to be precipitated. And precipitates means that we need to take it out of being liquid and form it into more of a solid material. So we need to be able to pull it out of the liquid and make it more solid so that it actually can be viscous. Viscous is a term that we use for something being very thick. It's going to form these viscous strands, and then we can actually spool it. What we need to do that is to add alcohol. And so ethanol is added, and ethanol is the actual component that causes DNA to precipitate, to come out of that liquid and be more of a viscous strand. So whether or not we're looking at a human cell or a plant cell or a cell from livestock, if we want to start with getting a copy of the DNA, we have to go through this process of DNA extraction. Once we have the DNA, then there's a variety of different types of biotechnologies that we can do depending on the goal of that particular process. What I'd like to do is just highlight two types of biotechnologies that have been used in labs over the last few decades or so, and that way we can understand more about how these technologies can help us with some of the things we mentioned before, like livestock and crops and medicine, etc. The first technology that I wanted to talk about is something called PCR, and PCR stands for polymerase chain reaction. So let's just start with the two words here that might be most familiar to us and think about what it means to have a chain reaction. If you think about what a chain reaction is, it just means that there's one event that causes another event that causes another event, which causes another event. And that's a chain reaction. These things are all linked to one another. And when we're talking about PCR, what we want to do is have a chain reaction occur, and we want it to be a chain reaction in copying DNA. So the goal of PCR is to start out with just a few copies of DNA and then to rapidly increase the number of DNA molecules. We want to make many copies, and we want to make it really quickly. And that's what PCR does for us. When we think about making a lot of copies, let's first think about some of the uses where this might occur, and then we'll go through the details of how to actually do PCR. You could imagine that if you were a police officer or a crime scene investigator and you went to a crime scene, that you might collect a very small amount of DNA. Maybe at a crime scene you only get just a few hairs or maybe a blood sample, and it tends to be very small. But you want to go back to the laboratory and do a number of lab experiments. One of the very first things that you would want to do is to carry out PCR so that you can make a lot of copies. You want to increase the number of copies that you have, and that way you will have enough of the DNA to do all the tests that you want and likely save some for later and for future uses. Another time that we might use PCR is when we find ancient DNA. So a few years ago, we found a woolly mammoth that was frozen in the ice, and we were able to get some DNA from it. Not a lot of DNA, a little bit. And so one of the first things we wanted to do was to be able to rapidly increase the number of DNA molecules that we had so we could do a lot of different types of tests on this woolly mammoth DNA. But PCR can be used for a lot of other reasons too. We know that we can use PCR as a first step in paternity cases. And anytime we're doing genetic testing, we likely are going to make a number of copies of the genetic material. When we think about how to do this, I hope that you actually know something about the process already. If you remember at an earlier point in our semester, we talked about the process of DNA replication. So it might be worth actually going back and reviewing that now, and then coming back here and pressing play again for our PCR. When we carry out our PCR then, we have to start with a few reagents. And once we put those reagents in a tube, we're going to add them to the machine that's shown on this picture. What you can see on this machine is something called a thermocycler. So a thermocycler is a machine, could look like that, could look like this. There's a variety of different ones. And they are a piece of lab equipment that generally sits on the countertop. And they do exactly what their name suggests. They actually will cycle the temperature. So it's almost like your thermostat at home. If you were to cycle your hot and cold over the course of a day, then this might do this over the course of a few hours. And it's going to create then a really hot condition and then cool things down, and a hot condition and cool things down, so that that's going to promote this chain reaction. And we're going to go through those steps. What you actually put into the thermocycler then is going to be a tube, many tubes, actually you can do a lot of these at one time, that's going to contain some important reagents. So let's talk about what you would have to actually put in your tube in order for your tube to be able to carry out this chain reaction. One of the first things that you'll need to add to your tube is going to be the original DNA. So maybe that's DNA from a crime scene or from a woolly mammoth, whatever that might be. The second thing that we need to add is a number of free nucleotides. So if we're going to make many, many copies of DNA, we need to have the raw materials to do that. So we know that we have to add a bunch of A's and G's and C and T's so that they're available when the process of DNA replication is occurring. So that's the second thing that we have to add to our tube. One of the other things that we have to add to our tube is going to be an enzyme. And that enzyme specifically is going to be the enzyme we've talked about before, which is DNA polymerase. Now we actually have a challenge with this particular enzyme. I hope you remember that enzymes, if we put them under really hot conditions, can be denatured. And so it took a little bit of troubleshooting, but we actually now will use an enzyme, a type of DNA polymerase, from a bacteria that is found in hot springs. If you think about a bacteria that lives in a hot spring, it has to be capable of carrying out DNA replication. However, these are animals that are well adapted to living at really high temperatures. And so those particular type of bacteria have these enzymes that don't get denatured at these hot temperatures. So the particular type of DNA polymerase that we use is actually called Taq polymerase. And that Taq polymerase, that word comes from the actual name of the bacteria, which is Thermus aquaticus. So if you take the T with the A and the Q, you're going to get Taq. So that's actually how this is named. We know that Taq simply tells us that this is the type of enzyme, and we get it from a bacteria. And that means that this can be heated up and we can use it in our tube. The final thing that we have to add to our tube is something called a primer, and usually more than one primers. Generally speaking, when we copy the DNA, we don't want to copy all of it. We just want to copy a portion of the DNA. And so the primers are going to indicate where we are going to start copying and where we're going to end. So it essentially brackets the DNA and says, copy in this particular location, but you don't need to copy everything. We're going to look at some animations of PCR, and you'll be able to see not only the enzyme at work, but also how the primers work as well. I'd like to go through an animation of PCR so that you can see how it works and how the thermocycler is going to be important in actually getting our copies of our DNA. For that reason, I'm going to go to a website that you can see here, and that is www.dnalc.org. And then once I went to that website, I simply just searched for PCR, and it brought up a series of choices, and then I picked this particular one here, making many copies of DNA. There's a lot of great videos and animations at this website, so you may want to take a look around to see what you can find. I'd like to go through this animation with you, specifically focusing right now on the amplification. This is going to show us a step-by-step guide for how DNA is copied in this PCR reaction. You have an image in your book that's very similar to what we'll see here in the process as we go through DNA replication. And so you'll see these steps highlighted in a static picture while we're going to go through the animation together. If we think about our very first step, the first step that we need to do is to heat up our DNA. So our machine, our thermocycler then, is going to warm up, and it's going to warm up to 94 to 96 degrees Celsius. So this is quite warm. And this is something that you would program into the PCR machine. What this is going to do is to denature our DNA, which I'll show you in just a second. But I want to make sure that we understand how important it is that we use Taq polymerase. If we took human DNA polymerase and heated it up to 94 to 96, we would find that it would actually denature. And so it's important at this step that we have the correct DNA polymerase. So if we take our DNA and we heat it up and we denature it, which we want to do, that DNA is going to actually separate. That's what we mean by denatured DNA, is it's going to separate. And then you can see our temperature is going to cool. We're actually now going to cool it down so we can add our primers. Annealing, that word there, anneal, simply means that we're going to add and attach the primers. And remember, our primers are there to make sure that we can highlight the specific area of DNA that we would like to copy. So let's add our primers, and you can see them coming in here, and one is going to be called an upstream, one's a downstream, and we know that they are going to attach to show where to copy. Now we're going to do something called extending the primers. Those primers then are going to be important for synthesizing the new DNA. So let's actually see the enzyme that comes in to do that. That enzyme is our Taq polymerase, and it's going to follow where those primers were, and you can see that it's made now new DNA, and we've got 2 copies. So right now we are essentially at the end of our first cycle. We started with 1 copy of DNA, and now we have 2 copies. Let's go to our second cycle. We are now going to heat things up again by denaturing our DNA. So we're going to separate all of that. Then we're going to cool back down so that we can add our primers, anneal them. And now we're going to get our enzyme involved, and we've warmed up again slightly to 72 to actually have our Taq polymerase do its job. And so it comes in, and now we're at the end of cycle 2. So at the end of the second cycle, we now have 4 copies of our DNA. I hope you realize that every time we go through a cycle, we are going to double it. So after our first cycle, we had 2. After our second cycle now, we have 4. So we could keep going. And you could see that through each cycle, we are going to add our primers, we're going to get our Taq polymerase working, and you can see that we're now producing more and more and more. Let's take a look at the amplification graph now. If we were to actually count how many copies of DNA we could get over just a few cycles, you could see that after 5 cycles, we would have 32 copies of DNA. And if we continue, and we're just looking at the red line here, now together with the purple line, you can see that just over a number of cycles, we could end up with 67 million copies of DNA, assuming we added enough free nucleotides to be able to copy all of this DNA. At the end of this whole process then, you would have a number of copies of DNA to do many different tests, as well as to store some in the freezer for later use. How long does all of this take? Oftentimes, lab scientists will set up their PCR machine at the end of the day, and they may tell it to go through 30 or 40 cycles overnight. And when the scientists come back in the morning, they now have all these many copies of DNA. So it takes a few hours, but it doesn't take many days. It's something that can be easily done in the course of a 24-hour period. The second technology that I wanted to discuss is gel electrophoresis. Gel electrophoresis allows us to compare DNA samples. So these might be DNA samples from 2 different people, and we might be trying to identify ways that they're similar, ways that they're different. We might have an unidentified DNA, for example, DNA at a crime scene, and we're trying to compare that DNA with some suspects to see if we have a match and can identify the individual with that DNA. So I wanted to show you a finished gel electrophoresis, and as we look at this gel, we might be able to see some similarities, and then we're actually going to go through the process of how we can actually run a gel. What you'll notice at the end when you examine this particular gel is that we have a number of bands that are going to be visible from our gel. Now, if we look very closely at some of these bands, you'll notice that some of these different columns, and so here's a column and here's a column. We've run a number of different samples here. Actually, when we look on the ends here, you'll notice that the ends have what's called a ladder. So this is a standard by which we put on our gel. But you can see that we've actually done about 7 different DNA samples in the middle of these 2 ladders. When you look closely, you'll be able to see that some of the banding patterns are similar and could indicate the same person, or at least the same genotype with respect to the gene that we're examining. So for example, this column here and this column here both have some bands right here that are going to be exactly the same, and so those are going to be similar in this way. We could also examine 2 other columns that are similar. This column here and this one here also have a similar banding pattern. We can also see where 2 columns may share one thing but not another. So if we look carefully at this column right here, you can see that there's one band. This column here also has that same band, which is right here, but there's another band above it, this one right here, that is not shared. So when we examine those 2 columns, you can see that there's some things that are shared and some things that are not shared. So the end result is that we can actually see whether certain genes or alleles are shared between our different DNA samples or not. And this can help us out, like I said, with identifying individuals or even determining paternity or taking a look at different children of parents to determine their offspring. We're actually going to use a gel electrophoresis process to finish up our case on MSUD. Before we do that, though, I want to talk about how do you get this? How do you actually make a gel based on what we've talked about so far? So I'd like to take a look at a picture that you have in your course manual so that we can go through the steps of how this actually would work. What you'll notice in the first steps is something we've already talked about, and that is we actually have to get our DNA sample. We've already gone through the process of how to extract DNA by breaking apart cell membranes, by adding enzymes to prevent DNA degradation, and then by precipitating the DNA with ethanol, with alcohol. So we're assuming at this point we now have a tube that has our DNA sample in it. The next thing that we need to do is to actually cut the DNA up into fragments. We need to make sure that when we run our gel electrophoresis that the DNA is in smaller pieces so we can have those pieces of DNA actually physically move through the gel, and we'll talk about how that happens in a minute. For right now, I want to talk about how it is that we cut DNA. The way that we cut DNA into fragments is by adding an enzyme, and very specifically it's called a restriction enzyme. Now, restriction enzymes do not just chop the DNA up randomly. They actually will target a very specific sequence. And so it might target AGCTTA, or it might target GCAACC. And wherever it sees that particular section, it's going to cut the DNA in that location. So you can purchase restriction enzymes that would cut the DNA in a certain place, and usually they're specific to the gene that you're looking at. So generally speaking, when we do gel electrophoresis, we don't necessarily want to run all of the DNA out in the gel. We actually just want to target a couple of places where we would have differences in DNA among humans, and we want to actually get a restriction enzyme that would target those particular places in the DNA, those particular genes. So before we go further, I want us to understand how we can cut this DNA and end up with fragments, and those fragments could have different lengths. Let's take a look at one of the activities that you have in your course manual. We're actually going to take these 2 individuals, and you can see that we've got person number 1, and we have 1/2 of their DNA double helix, and we have person number 2, and we have, again, 1/2 of their DNA double helix. So if we wanted to find out if person number 1 and person number 2 had the exact same sequence in a particular area, we could do gel electrophoresis to do that. Now, one of the things that you'll need to do first of all is to write down the complement of each of these. So wherever there's an A, you'll put a T. Wherever there's an A, you'll put a T, etc. And you have a place in your course manual where you can write that out. So please pause the video and do that now before we move forward. Once you've written your complementary base to each of the original DNA strands that we have here, the next thing we need to do is talk about where our restriction enzyme is going to cut. You can see in Activity 1 that our restriction enzyme is going to cut the DNA whenever it encounters a very specific sequence, and that sequence is CTTAAG. So what I want you to do is to take a look at person 1 and take a look at person 2 and see if you can identify where CTTAAG are located. And once you've done that, you'll want to put a bracket around that particular section. So let me show you an example. In this person number 1 right here, you can see CTTAAG. And if you wrote the complement to this, it would actually be read the other way as well. So for example, you can see that we would have CTTAAG. It should go both directions. So we're reading it this way, but the restriction enzymes will also read that way as well. So what I'd like you to do is scan through the DNA and find any of these restriction sequences that this enzyme might look for and simply put a bracket around them. So please pause the video and do that now. Find out if there are any other in person number 1 and if there are any in person number 2. Here is what you should have found with regard to the specific sequence CTTAAG. Each person has this sequence, but it's in a different location in this particular strand of DNA. Once you've identified that, we're going to recognize that restriction enzymes, like a pair of scissors, are going to cut through that. So you could imagine that we just cut through and cut that section out, and now we're going to actually create many different fragments because of this cutting. So the next thing that we need to do is identify how cutting our DNA at these particular locations has resulted in fragments, and we need to find out how big these fragments are. So if we look in person number 1, you can see by cutting right here, we're just going to completely cut this section out, we've created 2 sections, or 2 fragments. And so there's one fragment, and we're going to stop it right at that A before we start the CTT. And then this fragment over here starts at the A and it goes to here. So we have now 2 fragments where we had 1. So our restriction enzyme acted like a pair of scissors, it cut our DNA, and now we have 2 fragments. We now want to find out how big they are, and we're just going to quantify these in terms of how many base pairs they have. So if we count these up, A, T, G, C, etc., and you should do that, we're going to find that this is going to be 22 base pairs long. That's how big this particular DNA fragment is. And if we look over here on this side, if you count them up, and you should, we have about 16 base pairs. So for person number 1, we've been able to cut our DNA. We have 2 fragments. One of those fragments is a little bit bigger than the other. One is 22 base pairs, while one of them is 16. I'd like you to do the same thing with person number 2. I hope you can see that we'll have 2 fragments. And why don't you take a few minutes and count up how big these fragments will be, and then you can restart the video. I hope this is what you found for person number 2. Person number 2 will also have 2 fragments. However, because the restriction enzyme cut the DNA in a different location, we're going to find that one fragment is much shorter. This is going to only be 6 base pairs long, whereas this fragment here is going to be 32 base pairs long. When you add a restriction enzyme then to a DNA sample, it is going to cut the DNA into fragments still here in our tube. And so now we have these different sizes of DNA fragments in the tube. Our goal now is to take those fragments and actually run them out on a gel so that we could see and visualize these different fragments. That's really the whole purpose of a gel electrophoresis then, is to be able to take these different fragments and separate them from one another, and in separating them we'll be able to visualize them. And so that's going to be our next step. Now that we have our DNA in fragments, we're ready to run them out onto the gel. I have put another video in our eLearn platform for you to take a look at in terms of how to actually load this gel and how the equipment works and how you have to make up your various solutions. That video is only about 7 minutes long, and I highly recommend that you watch that. What I'd like to do here is to summarize how this process works and what you'll need to do to actually get the banding pattern. One of the things that we have to do first is to load the DNA fragments into wells. Wells are simply pockets that are now present in this gel material. So this thing right here is a gel. If you were to handle it, it would feel like a piece of really firm jello. And in that firm jello, we've actually put in like a mold where we could have these pockets or these wells. And that's what these black areas are right here. Loading the sample can be challenging. You have to use something called a micropipetter, which is like a really small dropper, and you have to get a little bit of your sample and you have to then put that dropper in this tiny little well and then inject your sample into that well. So that requires a little bit of practice. Once that gel has been loaded with the different DNA samples, and you would put a different sample in each row, so you'd put one here and one here and one here. Once you've done that, now we have to actually plug it in and introduce an electrical current through this gel. It's very important that when we introduce this electrical current that we set it up so that the negative charge is here very close to where we put our DNA sample. Now the DNA sample that's in here has a charge. If we remember, our DNA is made up of nucleotides, and those nucleotides have phosphate groups. Phosphate groups have a slight negative charge. If you introduce an electrical field through this gel and put the negative here, that's going to start to repel the DNA, and the negatively charged DNA then is going to start to move away from our negative charge, and it's going to move down here toward our positive charge. The goal in introducing this electrical signal is to encourage that DNA to start moving through the gel. As these fragments start to move through the gel, you could imagine that if we have different sizes, they're going to move at different speeds. So if we look at our last picture here, we actually find that large fragments don't move nearly as quickly. As these fragments move through the gel, it's just harder for larger fragments to get as far. So when we're separating these things out, you can see then the larger fragments are going to be closer to where we started. Whereas if you're a small fragment, you can move very easily through the gel and you will move much further. And so these bands down here are smaller in size. So when we look at how gel electrophoresis actually separates our DNA samples, it separates them based on two things. First of all, it separates our samples based on charge, and secondly, it separates them based on size. And that's what we want to do, is to separate these fragments. If we had two different DNA samples that had exactly the same banding, that could suggest to us that those two individuals are the same person. But it doesn't necessarily prove that they are. It just says that they have the same length fragments. However, if we see two individuals, two DNA samples, that have different banding patterns, we know absolutely that they are different individuals. I'd like to go back to our previous simple example that we talked about with our two individuals, person 1 and person 2, with our restriction enzyme. I want you to imagine that this is a gel, and we are going to run our samples out on this gel. And so we're going to load person number 1 here into this column or row. And we're going to load person number 2 into this column or row. I hope we remember that person number 1 had two fragments, and one was 22 long and one was 16 long. So let's put a couple of bands here for person number 1. Which is going to move further, the fragment that is going to be 16 or the fragment that's going to be 22? I hope we remember that the smaller ones are going to move further. So let's say we have a band here, and let's say we have a band here. And that means that this band was the one that had 16 base pairs. Because it's smaller, it would move faster. And this is the one that is going to have 22 base pairs. It's a little bit bigger, so it won't move as far on our gel. However, with person number 2, while we did have 2 different fragments, those fragments were different sizes compared to person number 1. So let's take this fragment by fragment. One of the fragments in person number 2 was 6 base pairs long. It was really short. It was a really small band. And so if we think about where we need to place that band, it's going to be roughly about right here. It's smaller than the 16, and so it will travel further than our 16. The second fragment in person number 2 was 32 base pairs long, so it's bigger than anything that we saw in person number 1. And so that band might be here, for example, not traveling very far at all, away from our pocket or our well. And that one was 32. So at the end, if we compared a gel from these 2 individuals, we would find 2 fragments for each. However, these 2 fragments would be in very different locations because of the size of each of these, and we would be able to confirm that person number 1 and person number 2 are not only are different people, but they don't share any of the same fragment lengths because they move to different locations in our gel. Now that we know how gel electrophoresis works, let's return back to our family and find out how performing a gel might help out Emma, Jacob, and Matthew. Let's first get an update on Matthew. We know Matthew is 3 years old, so we have fast forwarded quite a bit, and he's doing very well. We know that Matthew has maintained a low protein diet, and we know there haven't been any close calls, but Emma and Jacob are considering further treatment, in that they've decided to put Matthew on the waiting list for a liver transplant. Previously, we had talked about some of the pros and the cons for doing that. We now know that Emma is pregnant again, and for religious reasons, they have agreed that they do not want to terminate the pregnancy even if the baby is found to have MSUD. However, both she and Jacob want as much information as they can before their baby is born, and so they decide to seek genetic counseling. In order to do this, they took blood samples from Emma and Jacob and Matthew, and they also were able to get some skin cells from the fetus, from the amniotic fluid. Just as you shed skin cells every day, so does a fetus, and those skin cells can accumulate in the amniotic fluid. So in an amniocentesis, some of those skin fibroblasts can be obtained, and we can actually do genetic testing on the fetus to determine its genotype with respect to MSUD. Let's remember a few things about Emma and Jacob and Matthew regarding their genotype and their phenotype. I hope we remember that Emma was a carrier for MSUD. Otherwise, she was healthy, but she did have a heterozygous genotype. And Jacob was the same. He was also healthy with a heterozygous genotype. Matthew, on the other hand, is little m, and we know that he has MSUD, this recessive trait. If we think about the chances that Emma and Jacob will have another child with MSUD, we can simply do a Punnett square. If we do a Punnett square, we can place Emma's genotype on one side and Jacob's on the other, and we can fill in all of our offspring and realize that there is a 25% chance that the offspring will have capital M, capital M. There's a 50% chance that, like their parents, they will be heterozygous, and a 25%, like Matthew, that the fetus could be little m, and have MSUD. What we're going to do is do a gel electrophoresis with some of our DNA from our family to determine what the genotype of the fetus will be. We need to understand very carefully how our restriction enzyme works in MSUD before we actually run this out on the gel. So let's make sure we take a look at this information. We know that we're going to take Jacob, Emma, Matthew, and the fetal cells, and we're going to add to each of those samples a restriction enzyme. The restriction enzyme is going to cut our DNA only if we have a normal copy of the gene. So if we read carefully, the normal gene, and if we're talking about the normal gene, we're talking about the normal allele. So this represents for us capital M. It does contain the restriction site, And that means that if we have a normal gene, so let's just draw a segment of DNA, and then we add our restriction enzyme, so we'll add our restriction enzyme, that it will cut it into two smaller pieces. So let's imagine that we cut. That means that we should expect to see two fragments, and these two fragments would be smaller than if we actually had one big fragment. So if we were to have an individual who has a capital M in their genotype, we would expect to have two small fragments at least run out on the gel. The second copy of the gene has a mutation. That's going to be our little m allele, and this does not allow the enzyme to cut the mutated gene into smaller pieces. So when we look here, we would have a gene, our little m allele. We would add our restriction enzyme, but there would be no cutting because the mutation occurs in the site where the restriction enzyme should cut. If there's a mutation there, then the restriction enzyme doesn't recognize that sequence, and therefore it won't cut it. So what we'll have here with our little m is going to be only one fragment, but the fragment is going to be quite large. So just to recap, for this normal gene, if we run it out on a gel, we're going to have two fragments, and those two fragments are going to be smaller than this one fragment here that's going to be quite large. I want you to think about what we would expect to see for this banding pattern, thinking about how far large and small fragments actually move and what we would expect to see for Jacob and Emma. Once you've given that some thought, let's actually look at the results together. Let's take a look at Jacob and Emma. We remember that Jacob in his genotype is going to have a copy of capital M and a copy of little m. Let's remember what that means in terms of our banding. For our capital M, we know that that allele is going to break into two smaller pieces of DNA. So if we remember that larger fragments are going to be on this end and smaller fragments are here, these two bands right here represent capital M, because our capital M gets broken into two smaller pieces, and smaller pieces are going to travel further. But we also know that Jacob carries in his genotype a little m, which means that there's going to be one large fragment that doesn't travel very far that represents the little m. So this right here, this band, represents for us little m. So anybody for MSUD who has a heterozygous genotype should have their capital M represented here and their little m represented here. Emma would have the exact same banding pattern. Even though they're separate people, we're only looking at the genotype for MSUD. Therefore, Jacob and Emma should have the same pattern. Let's look at Matthew. We know that Matthew is little m, which tells us that the second copy should not break into two pieces. So he only has one band here representing the little m. In fact, he probably has two of them. They move the exact same distance, and they're probably sitting right on top of each other. By not having any bands here, it tells us that he is missing the capital M. Let's take a look at our fetus and see what we find. I hope when you look at the fetus, you can see that this is very good news. What we notice is that the fetus does have the two bands that represent the capital M. In addition to that, you can see the absence of a band here, which means that the fetus is missing the little m. So we can tell from this gel that the fetus's genotype is going to be capital M, capital M. Homozygous dominant, the fetus is going to be healthy, and the fetus will not be a carrier even for MSUD. I hope you've been able to see how this particular gel has answered this question and has been used as a diagnostic tool in telling us the genotype of this fetus. What I'd like you to do in the next activity is something that we've done previously, where you carry out transcription and translation in order to figure out what type of mutation we have in MSUD. Please use the codon dictionary that we have used in a previous exercise to complete the process of translation.